Now, our first speaker today is Dr. Amanda Hudson, who is representing Flagship 1. Thank you, Amanda.
Thanks everyone for joining us today. So I am the Cancer and Inflammation Flagship Fellow, and I'll just be giving you a bit of an overview of my project, which is entitled Modulating the Inflammatory Responses to Improve Treatment Efficacy in Brain Cancer. So first of all, the cancer I work on is glioblastoma, and this is the most common primary brain tumour in adults. It's extremely aggressive, invasive and lethal, and the median survival is just 15 months. Standard of care therapy consists of surgery, followed by radiotherapy and temozolomide chemotherapy, but unfortunately this isn't curative, and this means that progression is inevitable, and this therefore means that new and more effective treatments are urgently needed. So recently the importance of the immune macrophage has become apparent in glioblastoma, but also in many other cancers. So the first point to note here is that there's actually a lot of them in the cancer, and they can actually make up 30% of the tumour bulk. Secondly, rather than actually identifying and killing the cancer cells, which is what immune cells are meant to do, the macrophages are actually coerced by the cancer, and they actually create this environment which is beneficial for it. So they're able to stimulate tumour growth, release factors which can also form blood vessels, and also are able to enhance tumour invasion. Additionally, they're also recently been shown to play a role in tumour recurrence and also in therapy resistance. And it's this therapy resistance which is what this project is all about. So the hypothesis of this project is that tumour-associated macrophages, or TAMs for short, are able to help protect the glioblastoma cells from treatment-related death, and therefore any new therapies must be able to inhibit this glioblastoma macrophage relationship. Now the aims of this project, so this is a basic research project, and the aim is to identify new therapies that are effective against treatment-resistant glioblastoma by inhibiting that crosstalk that occurs. So in terms of the method that I'm going to be using, it's a co-culture system where I grow treatment-resistant glioblastoma cells together with the macrophages, and then I'll be screening a novel compound library. Now this library actually consists of compounds which are predicted to cross the blood-brain barrier, which is obviously important in a tumour which is located in the brain. So the compounds that are effective in inhibiting the crosstalk or causing cell death will be identified, and a hit will be classified basically as anything that's able to kill 50% of the cells, and then these will be examined in more detail. And just over on the left-hand side, you can just see the stages. It's sort of broken into the initial screen, then second stage is confirmation of the hits, and third stage is examining these compounds in additional models. Now, as I mentioned, I'm using a co-culture system, and one of the cell types is a glioblastoma cell line that's resistant to temozolomide. And this curve here just shows you that it's resistant. So on the x-axis, you have increasing concentrations of temozolomide, and on the y-axis, you have the percent survival of the cells. And in red, you can see these are the temozolomide-resistant cells. And basically, this is just a kill curve, and it shows you the concentration that's needed to kill 50% of the cells, or the IC50 value, which is that dotted line there. And as you can see here, the cells in red are actually sevenfold higher, or they need a sevenfold higher concentration of the drug, which means they're sevenfold resistant to the drug. So these will be used together with the macrophages in the co-culture system, and that's just a picture of what they look like. And I've also transduced the glioblastoma cells so they're green, so you can distinguish between the two. So the first experiment I performed was sort of a proof of principle to ensure the co-culture system was working properly, and also to help prove the hypothesis that the macrophages are actually protecting the glioblastoma cells. So to do this, I've added in a compound into the co-culture, which is known to downregulate the ability of the macrophages to protect the glioblastoma cells. And then I've performed another cytotoxic curve, or one of those kill curves, and looked at the response. So the curves here in the orange is when you add the compound, which is able to downregulate the ability of the macrophages to protect the glioblastoma cells. And the fact that it's moved to the left in comparison to that black curve means that the glioblastoma cells are now more sensitive to the drug, and therefore the macrophages are helping to protect them. So we now know that the system is working, so I could move on to stage 1 of the project, which is to screen the 1,000 compounds. I've done this at 1 dose to start with, and it's 10 micromolar in duplicate. And this is just a heat map to help visualize the data. So the 1,000 compounds are spread on 17 different plates, and there's 60 compounds per plate, and each of those little boxes represents one compound. And basically in the dark red is compounds which result in 0 to 20% survival, so they're very potent. In the lighter red is 20 to 50% survival, and in the grey are ones that are not particularly effective. So it's the ones that are in the red colours that we're going to be moving into stage 2. Now this stage is still in process at the moment, and basically it's just to identify those compounds which are the best, and we do that again by using those kill curves. So we want the compounds which have that lowest IC50 value, so any curves that are more to the left are the best. And these curves here are just examples of 4 compounds on each to show you that, for instance, in the one on the left, the red and black compounds are most effective, and the one on the right, the one in the dark blue, is the most effective. So I think out of 92 compounds I found in the first initial screen, I've screened 72 in this confirmation stage, so I've still got a few more to do. But in terms of what comes next, we'll be testing them together with temozolomide and radiotherapy, and then any that we find to be the best will then move into the in vivo models. So the last slide is just, if it comes up, a slide I was asked to put in, which is a measure of success. Obviously, this is all based in the lab, so due to COVID, obviously it's been quite delayed. But regardless of that, I've been able to publish 4 different papers. Two PhD students that I supervise have had their theses accepted, and there's another one currently under review. I've been able to secure 2 grants, and also I've presented at another conference. So just brings me to thank everyone for coming, and thank everyone who's helped. Thank you very much.
Thank you.